Hello and welcome to the International IVF Initiative, live session 6. We have around about 1,300 registrants for today's session, and so we're going to wait a little while until most of them have managed to join us. We're now up to 500. Just a few words about where we are today. The COVID-19 pandemic has really initiated an international crisis with a devastating impact across the world on the way we work and interact. In spite of the promising downward trends we're now beginning to see in Europe in terms of the numbers of new cases and deaths, it's going to take us quite some time before we can really turn the corner and return to normality. Social physical distancing is a critical component of our response to containing further transmission, leading to isolation, which can be very, very difficult. Isolation from friends, colleagues, and even family. We have therefore coordinated a global collaboration team, including now 14 organizations, to bring reproductive professionals from around the world a unique educational and social program. Here are the 14 organizations which have joined our cause. I'd like to thank them very much indeed. And also just to say that this is a non-for-profit organization. We'd be very happy to receive any donations from individuals. And coming back to the format of today, just to inform you that all attendee audio video will be turned off for the entirety of the session. We would request you please to use the Q&A button, which you can see on your screen, to ask a topic-related question to the panelists. Please try to avoid asking questions which have already come up. These will be filtered by our panelists in the background, who will then collate the questions so that we can actually give those live to each of the speakers. Just want to introduce you to some of the people who are working in the background. The I3, or International IVF Initiative team, have already coordinated 4 virtual sessions here at ivfmeeting.com over the past 2 weeks. And we will continue to develop a series of live educational sessions, as well as tutorials, panel discussions, and other activities. Now, from last Friday's session on SARS-CoV-2 and IVF Revisited, the team here at I3 are actively working on the Q&A document, which will be uploaded, and it should be online by the end of the week. Now, in order to make all of this happen, we have a tremendous team behind the scenes working to deliver an excellent event. And I'd really like to acknowledge in particular our Zoom tech support, Marianne and Thomas, who do a great job in making sure that the platform is working and that we're all doing what we should be doing as panelists and speakers. But also I'd like to say, a thank you to all those who have participated so far in the sessions. And it's my great pleasure in particular to actually be the host of today's session, which is on in vitro growth, IVG of human ovarian follicles, future developments. My name is Colin Howles, and as you can see from my flag in the background, I'm based in Geneva, Switzerland, which is the home of the Red Cross, and I'm a member of the I3 collaboration. So here is the program for this evening. In vitro growth of human ovarian follicles, future developments. We have two excellent speakers, and Professor Richard Anderson, who is the academic moderator. He will have the task of putting everything together and making sure that things flow between the two speakers. Just before we actually move on to the program, I just wanted to remind people that there will be a further session on Friday of this week. The next session is an introduction to quality control, part 1. This is organized by NIMES and Start Embryology Societies. And we have a list of panelists taking part, excellent people who really know what they're talking about. Eva Schenkman, Anthony Anderson, Carol Kirchhoff, Thomas Rusty Poole, and Giles Palmer. So, without any further ado, I will come back to today's session, and I will now hand over to the academic moderator, Professor Richard Anderson, who will introduce our speakers. Thank you. Richard.
Thank you very much, Colin, for that introduction, and welcome to everybody who's watching and listening today. Just fantastic initiative on behalf of this group to pull this together, so congratulations to the team for doing so. So, as Colin has indicated, our theme tonight is in vitro growth of human follicles. So
immediate pH and CO2 concentration.
The ordered growth of ovarian follicles from the primordial stage through to ovulation and the production of a competent gamete is absolutely central to reproduction, and we deal with it every day. And yet it remains really surrounded in mystery. We have little understanding of how the ovary makes its stock of follicles last for nearly half a century. And yet at the same time, for about 35 years, it can produce a mature oocyte every 28 days. And much of what happens here is pretty invisible in vivo, so the development of in vitro techniques is essential if we're going to advance that knowledge in the subject. And we have two great speakers to bring us up to speed on where we are with this at the moment. So our first speaker is Professor David Albertini. David's currently senior scientist at the Center for Human Reproduction and the Rockefeller University in New York, and consultant at the Bedford Research Foundation in Massachusetts. He's also the long-standing editor-in-chief of the Journal of Assisted Reproduction and Genetics. So David brings a lifetime of experience and enthusiasm in oocyte biology, and he's got particular interest in the communication between the oocyte and its somatic environment. So he's going to kick off our talks today, giving us a background into the history and the ongoing challenges surrounding the in vitro growth of ovarian follicles. So David, we look forward to your talk, and over to you.
Thank you, Richard. It's a pleasure to be with all of you today. I would also like to acknowledge the kind invitation to participate in this program. Thank you to all of the organizers for making this possible. Theme for today, the in vitro growth of human ovarian follicles, is one that has been of great interest for many years, especially in the world of animal reproductive physiology. But it's really only been in the last decade that we've been able to apply and further develop some of these techniques to go forward and make an impact in the utilization of this technology for human health. I note here just my disclosures, and would like to move on to now remind all of us of the extraordinarily extensive supply of primordial follicles, as illustrated here. This is a slide that was kindly provided by Professor Klaus Anderson in Copenhagen. And it illustrates for you something that Professor Anderson just mentioned, which is the the profuse accumulation of primordial follicles within the cortex
Excuse
of the
me,
human
David?
ovary.
So sorry
Hello?
to interrupt. We can't see your screen. Could you share it?
Oh. Where'd it go? Aye. Okay, here is the disclosure slide. And the slide that I was referring to was this one, which illustrates the distribution of primordial follicles within the human ovary. This happens to be, as I mentioned, a slide obtained from Professor Anderson, and it illustrates the expression of GDF9 in the primordial follicle pool of a 7-year-old patient. To the right is an electron micrograph of the human primordial follicle. And it really sets the starting point for the process that we're going to be talking about, which is a process that takes many days to bring the oocyte and its surrounding follicle to the state of maturity that would allow for both the resumption of meiosis in response to the LH surge as well as ovulation. Okay, I think to get started, what I'd like us all to bear in mind is that the ovarian follicle is both an exocrine and an endocrine entity. And as much as we have studied the process of follicle development and oocyte development as separate entities, we've now come to understand that these processes are inextricably linked. And that, as Professor Anderson just mentioned, this coordination between ovarian follicular development and oogenesis is essential for the production of viable oocytes from which future generations will be propagated. It is also very clear now that the follicle serves a number of very specific functions with respect to the oocyte. It sustains the growth phase of oogenesis, that is the growth of this very unusual cell. It supports the metabolism of the oocyte directly, and it regulates the cell cycle status of the oocyte, which is so important at the time of the LH surge, when the meiotic cell cycle will be reinitiated. And finally, the follicle serves an important endocrine function, as we know, establishing the output of estrogens during the follicular phase, and following ovulation, it transforms into the progesterone-secreting corpus luteum, and therefore plays an essential role in coordinating progress in the menstrual cycle. It's helpful at this point to consider a bit of the history, because the history in the field of in vitro follicle growth really underwent a shift in the 1980s. Up to that point, as shown on the left here, it was more the endocrine properties of the follicle that were the subject of interest, led by workers such as Gilbert Greenwald, who used the hamster as a model. And it was also during this time that a number of groups working with non-human primates began to study the behavior of the follicle in isolation, especially with respect to the control of its steroidogenic activity. But what we have seen from the 1980s to the 1990s is a dramatic shift from studying the follicle as an endocrine entity to one that could undergo ovulation in vitro and provide, under those circumstances, oocytes would be capable of initiating and sustaining embryonic development. And really, the leader behind this effort was the laboratory of John Eppig at the Jackson Labs in Bar Harbor, Maine, who during the 1980s and 1990s established a number of techniques that we now take for granted in the human ART field, whether this be in vitro maturation, in vitro fertilization, variety of techniques that really opened our eyes up to the basic biology of mammalian development. These were pioneered by Eppig and his colleagues, and during the 1990s, a number of workers, including Rita Kortfriend and Johann Schmitz, were able to refine these models to the point where both ovulation induction and production of quality eggs, quality oocytes, was produced in an effort to show that in vitro growth of follicles could be attended by the complete development of the female germ cell. Joanne Fortune explored these studies in the bovine model, and our second speaker today, Professor Telfer, really took the work of Eppig to a new level with different species, and as you'll hear today, has extended that work to the human. It's important in considering the history of this field that we keep in mind the concurrent development of the field of fertility preservation. Because it was with the onset of this new area, this new specialty area in reproductive medicine, that the materials that would be needed to develop this technology really became available for the first time. And as we look to the rationale behind this effort, both historically and contemporaneously, we're led first to understand that as a basic science problem, the complexity of follicle development and the enclosed oocyte has been very difficult to assess in the intact organism. So the impetus has always been to reduce the physiological complexity that is apparent within the reproductive system. We've also seen over this time the importance of understanding how the primordial follicle pool is established and maintained and modulated through the reproductive lifespan. So understanding what regulates the activation of these follicles. And it's also been a remarkable decade in which a number of paracrine and autocrine factors produced by the ovary, by various compartments of the ovary, have come to be recognized as key elements in controlling the development of the follicle in vivo. And that, in fact, has led to the definition of various media supplements that, as you will hear later, provide for multi-step models for sustaining follicle development under in vitro conditions. And here I illustrate with the diagram the concept of dynamic reciprocity, which really has been enabled as a result of the studies in vitro follicle growth. And it has exposed a remarkable feedback system between the oocyte and the granulosa cell with respect to specific signaling molecules and receptors that titrate the growth and differentiation status of both the oocyte and the follicle to subserve their respective needs. And finally, before we move on, I'd like you to understand that this dialogue between the oocyte and granulosa is just one component, is just one aspect of this in the follicle, and that we need to keep in mind, as shown below, that really there are three critical cell types that monitor each other with respect to their state of differentiation, that being the theca, the granulosa, and the oocyte itself. Facing the challenge of propagating in vitro growth of the follicle has meant that we also have to face the biological imperative, which occurs once primordial follicles are activated and initiate their growth phase. The initiation of growth, which involves, in the case of the oocyte, hypertrophy, and in the case of the granulosa cell, hyplasia of the granulosa cells, will be followed by phases of maturation, ovulation, and then, as we said, the transformation of the follicle into the corpus luteum. And along this pathway, follicular atresia is not only a dominant force, but it is, in fact, behind the attritional process that accompanies reproductive aging in the female and results in the gradual loss not only of the follicle reserve, but the diminishment in quality of oocytes seen with advancing maternal age. So trying to begin to understand how these factors signal not only key transitions in the follicle, but maintain the livelihood of these under in vitro conditions has been a particular challenge, and one that we're facing today in the studies that we're undertaking. How long does it take the follicle to develop and produce a developmentally competent egg or oocyte? We're really reliant at this stage of the game on the original estimates made by Professor Goujon that the process from the time of primordial follicle activation to that of ovulation and the resumption of meiosis would encompass several menstrual cycles, approximating perhaps 100 days. This is guesswork at best, because the dynamics of follicle growth are probably far more complicated than we currently appreciate, but 100 days is approximately the amount of time that we think it takes to reach this point in development. And as you will see from Professor Telfer's talk, many of the events that we are trying to recapitulate under in vitro conditions probably take place over a much shorter time interval. But there are some common principles that have been gleaned from understanding the physiology of follicle activation, growth, maturation, and ovulation in vivo that we have done our best to apply to an understanding of this system under in vitro conditions. These include initially the importance of establishing the soma-oocyte interaction that would both maintain quiescence or propagate an activation stimulus to launch the process of folliculogenesis, that this process is nurtured within the ovary by the follicles developing a rich vasculature that will sustain their growth and secondarily that of the oocyte. And critically, as we will see, there's the establishment of a bidirectional communication pathway between the oocyte and the granulosa cells, and we'll come back to this image shortly, that finally allows for the execution of competence in the female germ cell. Now, what do we mean by competence? What we mean is that the female germ cell acquires over the course of this journey a series of competencies that will endow it with the molecular and biochemical components needed to initiate meiosis, complete meiosis, initiate fertilization and egg activation, and sustain development all the way to the point of implantation. And this dynamic reciprocity, okay, really illustrates for us the importance of these dueling interests in development of the follicle. What is the identity? Is there an identity crisis here? Is the follicle, in fact, being promoted as a structure that will sustain and support the development of the oocyte? Or is it, in fact, something that is being designed to transform into the corpus luteum should fertilization and implantation have taken place. And the interface between the oocyte and granulosa cell is really what allows for both of these functions to be subserved simultaneously during the course of follicle development. And I illustrate for you here a comparison of an early antral bovine follicle. These are from studies done in collaboration with Professor Telfer's lab. And on the left, you see the appearance of the gap junction network in green distributed throughout the follicular epithelium at the time of isolation of one of these follicles. And note the adherent thecal cells on this that will become relevant in the next talk. And to your right is a comparable follicle that's been cultured for 48 hours, in which you can see a shift to a more asymmetric distribution in not only the contour and distribution of the gap junctions, but also with respect to the oocyte itself. You can see that its cortical organization has been altered. And this is in fact the first of many challenges that we have addressed in the course of in vitro growth studies. And it bears directly on the conversation of the follicle is a three-dimensional structure. What can we do to preserve these three-dimensional patterns of organization, especially as extended culture platforms become more used? So in the end, when we talk about the follicle life cycle, we are talking about a very well-known series of changes, especially from the point of primordial follicle activation and the simultaneous growth of the oocyte in the follicle through the so-called pre-antral or gonadotropin-independent stages of follicle development, which then is followed under the direction of FSH by the gonadotropin steps in follicle development that will ultimately lead to ovulation. And of the challenges that have been recognized in the course of various studies in this area is the question of how to sustain this communication system in a target that is moving from one stage to the next. And we've learned much about the nature of the repatterning of these communication systems and their complexity, because there's much more than a gap junction based system to coordinate and synchronize cellular activity within the follicle. As you can see in the right, there are multiple forms of cell communication between the somatic cells of the follicle and the oocyte itself. And these forms of communication, whether they're driven by receptor tyrosine kinases or cell-cell adhesion interactions, are changing over the course of development. And this stands today as a singular challenge in the course of the kind of work that you're going to be hearing about. And so it does come down to the essential role that somatic contact plays in coordinating the dialogue between these and between these two discrete cell types, both for the benefit of the oocyte attaining its developmental competence, as well as for the benefit of the follicle sustaining its endocrine properties. Now, there's been much interest in this area, again, over the last 8 to 10 years, in the prospect, this is in the spirit of regenerative medicine and rebuilding everything from stem cells, that perhaps follicles and oocytes could be constructed from stem cells. And there have been some very significant strides that I would predict are going to ultimately play into the in vitro growth platforms that we're currently using, either as a supplemental or primary determinant in making this work. And so there's been much press about this work. It really started with the incredible paper in 2012 by Katsuhiko Hayashi, first showing that oocyte-like cells, oocytes could be derived from PGC-like cells to produce live offspring. There was the breakthrough work of Morihaku and their colleagues in 2016 that really began to lay down a foundation using principles from the field of in vitro follicle growth to elaborate more sophisticated systems with stem cells that allowed for the birth of mice. And as some of you may have seen recently in Science, the work of Nagaoka et al. Has finally broken that barrier of taking PGC-like cells through the actual meiotic process on the way to establishing oocytes from stem cells. So these are all exciting areas, and I just wanted to show you again. This is a very nice paper taken from Morihaku recently, and it summarizes for us on the top part of the chart the in vivo sequence of events from PGCs to oogonia and primary oocytes, and where those steps have been recapitulated under in vitro conditions. Again, this is all in the mouse at this point. And you will see, as I mentioned before, that the success in producing live birth from oocytes generated in vitro, again, harkens back to these earlier studies from Eppig and O'Brien and others that really set the stage for producing oocytes from in vitro cultured follicles. And this is just a slide that highlights two of the more essential steps that were identified in the study by Morihaku from 2016. The first, very early in the process, having to do with the breakdown of the germ cell cyst into individual primordial follicles that required the introduction of an estrogen receptor inhibitor. And then secondarily, the importance of modifying the connective tissue environment in the basement membrane of those follicles to get them to be able to sustain growth of the oocytes in vitro. All of these experiments, again, relying on in vitro maturation, in vitro fertilization, and embryo culture along the pathway to generating live mice. Very exciting work indeed, and of course, this is the stage that currently many people are trying to recapitulate this kind of approach for producing human oocytes. So this is not a case where social distancing, well, this is certainly a case where social distancing would interfere with the relationship that's established early on in the follicle that will sustain the simultaneous growth and development of the oocyte and follicle, and brings us to the close of this of my presentation as we consider, first of all, that oogenesis and folliculogenesis, as I've mentioned, are integrated processes, and they mediate in a reciprocal way the expression of both oocyte support and endocrine function of the follicle. This coordination we have seen is mediated by a very complex communication system that changes in form and substance at specific stages of development. And that achieving in vitro gametogenesis in the human will require examination of these parameters of the dialogue in order to obtain here, in this case, which you'll be hearing about, the first reports of human oocytes being able to initiate and achieve a maturation state as a result of the work that you'll shortly be hearing. So finally, acquisition of the competencies that will drive meiosis, fertilization, and implantation, we must remember, are all processes and abilities acquired within the ovary as the follicle develops. And more than anything else, these models are providing us with an opportunity to begin to unravel some of the sophisticated mechanisms that will underscore this so fundamental process in human reproductive biology. Thank you for your attention.
Well, thank you very much indeed, David, for that absolutely lovely and beautifully illustrated overview, giving us a great snapshot of the history. I always love it when people recognize that we grow our work delivers only because of what has gone before us, and also highlighting where the real state of the art in our understanding of these intercellular interactions, and as you highlight, how much of a dynamic process it is, changing as the follicle grows from one stage to the other over perhaps a very uncertain period of time. I think the plan is to have perhaps a question before we move on to Evelyn's talk. So I've got a list of the questions that have been asked here. Maybe one of them I think is particularly general is you had a slide that divided follicle development into the gonadotrophin independent and the gonadotrophin dependent phase. You know, right. Perhaps a bit of a black and white demarcation there. But what do you think that really, the relevance of that, the gonadotrophins and particularly obviously FSH, are to how essential is it for follicle growth in vitro, or do we really not need it in vitro?
Right. Well, I do agree with you that the the figure sort of is our historical representation of an on-off switch, and that we go from an independent to a dependent phase, and reality is very different. Okay, this is very much, I think, the more appropriately represented as a gray zone. Okay, that there would be a gradual transition in sensitivity. And we're beginning to see that, especially with respect to FSH, expression of the FSH receptor, et cetera. But I think as your work and that of many others has shown, this is not a simple gonadotropin on-off switch that is regulating that transition from the preantral to the antral stage. Certainly, we've all come to appreciate the importance of AMH over that transition period, and now there's a great deal of interest in how those follicles that reach a state where they are actively producing AMH, that this may in fact feed back on the primordial follicle pool and be another way that the ovary, using its own intra-ovarian paracrine system, is modulating movement in and out of the primordial follicle pool. As far as are we manipulating this with our in vitro growth systems, I have to say in general, we really haven't applied some of this knowledge to the in vitro growth systems to date. Evelyn will speak of some important experiments that her laboratory did with respect to the inclusion of activin in the medium, and that made a big difference. But I think it's still a very open area, and I think as we have learned more about the expression of these various peptides and hormones, et cetera, that we need to put them into the context of the IVG system.
Thank you very much, David. So can I remind the audience that after Evelyn's talk, we're going to have a broader Q&A session for both speakers. So please carry on putting your questions in through the Q&A button. So it's my great pleasure to introduce our next speaker, my friend and colleague, Professor Evelyn Telfer, also at the University of Edinburgh, where she holds a chair in reproductive biology. And Evelyn also brings many years of experience and expertise in follicle biology, which is studied in a range of different species, from rodents to livestock, as well as increasingly in the human. And indeed, her lab has been the only one in the world that's been able to successfully develop a method for growing human follicles all the way from the primordial stage through to the generation of a mature metaphase 2 oocytes. So I'm delighted to introduce her to give her next talk, our next talk on in vitro growth towards clinical application. So Evelyn, over to you.
Thank you, Richard. That's a very kind introduction. And I'd also like to thank the organisers for putting this programme together in these really difficult times. So it's really good to be contributing to this. So I'm going to be talking about the in vitro growth, and David has given an excellent presentation, and we've heard so much now about the oocyte and the requirement for the oocyte to interact with the somatic cells. So I'm just going to remind you about the stage of development that I'm going to be talking about the primordial follicles. So as you know, the primordial germ cells migrate to the gonadal ridge, and then they start to form interactions with somatic cells. And at that point, then there's the nest breakdown, and the oocytes, they enter meiosis. So they enter prophase 1 of meiosis, and they get to the dictate stage. And at that point, they are arrested. And they're forming these associations with the somatic cells to form the primordial follicles. And in the human, this is all happening before birth. And then after birth, there's utilization of this primordial pool. Now, the primordial follicles form a pool that won't be added to, and so there has to be something regulating the initiation of growth from that quiescent state into the growing state, where the oocyte becomes developmentally competent during this whole process of follicle development, which David outlined. And the somatic cells are becoming competent to produce the sex steroids required for maintenance of the cycles, the reproductive cycle. So eventually there's the antral follicle with the oocyte surrounded by the cumulus cells, and then the murogranulosa cells lining the antral follicle. And these primordial follicles, as I said, are a fixed population. So they're formed before birth, and at birth there's many of these eggs. And then by the time the girl reaches puberty, then there's substantially less of these primordial follicles. So there's a loss of primordial follicles with age, and particularly early on, there's a dramatic loss. And then again, in adulthood, there's less of these primordial follicles. And eventually, just before menopause, there are very, very few primordial follicles left. So the female's reproductive system is really vulnerable. The oocytes could be affected by many, many factors, and particularly chemotherapy treatment. So we now know that many therapies affect the number of these follicles, which can lead to infertility. And that has led to people looking for solutions to preserve fertility in women that are undergoing these therapies that might affect the primordial population. And the whole field of fertility preservation was developed in Edinburgh, and initially by Roger Gosden and David Baird. It was a really nice collaboration between a basic scientist, Roger, and the clinician, David Baird. And they started to look at removing ovarian biopsies, initially from sheep, where they would take small pieces of the ovary that contained many primordial follicles, and then they would freeze these biopsies, and then they would transplant them back. Now, that was the initial work was in sheep, and then they quickly moved towards a clinical application of taking these biopsies and preserving them in women prior to chemotherapy treatment. And now we know that what we can do with these human strips, so these are removed and frozen, and then when the woman, the patient who has undergone the chemotherapy, when she's well enough, then these strips can be transplanted. And throughout the world now, we have you know, this is a number that's changing all the time, but it's well over 150 live births from this methodology. And that's a primordial follicle population that's being stored and then restoring fertility in these women. But there's a problem with some of these strips, because many of the women who are storing these strips, they have blood-borne cancers, and some of the strips may well have malignant cells. So you couldn't really transfer them back, because you would have the problem of reintroducing the cancer that the woman has recovered from. So that has led to the search for methodologies to use the follicles that we have in the strips, but to avoid the transmission of malignant cells. And there's a group, Jacques Donné's group, Marie-Madeleine Dolmans in Belgium, have been looking at this whole process of artificial ovaries, where they remove mainly the growing follicles from these strips and try to get rid of any other cell types, and then put them in a matrix. And the idea is that they would then be transplanted. But of course, you still have the problems of, you know, are you absolutely certain that there are no malignant cells present? So that's led us to look at the whole process of lab-grown eggs. And there's other groups. There's groups in Chicago, Teresa Woodruff's group, and there's other groups in the UK and in Europe looking at lab-grown eggs. But we started looking at lab-grown eggs many years ago, initially with the mouse model, and then moving to domestic species, and now our focus has been on human. So the idea is, if we can grow these oocytes all the way from the earliest stage of development, then we could do everything in vitro and we would avoid any transmission. So it would be very useful for a particular patient group. I just want to emphasize then that Edinburgh started this whole process of tissue freezing in 1994, and now there's centres worldwide. And Richard Anderson, he heads up the fertility preservation programme in Edinburgh. So with that collaboration between Richard and I, we have access to a lot of material, human ovarian material, and I'll tell you more about the sources that we have. But if we just look here, you can see the image here of the histological sections of the ovary, and you can see that it's predominantly primordial follicles that are in these strips. There's some growing follicles, but it's mainly primordial follicles that are in these. So the challenge for us then is to develop these immature eggs all the way through to fertilization. So lab-grown eggs, David nicely put, they told you the background of the mouse models developing from the primordial follicles. So this whole process of in vitro gametogenesis or in vitro growth is the development of these, you know, early stage of follicles to maturity entirely in the lab. So complete in vitro development of the immature oocyte. But it can also refer to the formation of new oocytes from stem cells. And David spoke about that briefly as well. I'm not going to talk about that at all. I'm just going to focus on growing human oocytes from primordial follicles. So this is just showing you, and David spoke about John Eppig, and John Eppig really developed one of the best systems for the mouse model to get oocytes to grow all the way in vitro from the early stages. And this is showing you Egbert, who was the first mouse that was born from an in vitro grown primordial follicle using a two-step culture system. And there were some problems with Egbert, but this highlights that by modifying your culture system and in optimizing it, you can then improve your results. And that's exactly what John Eppig did. And in 2003, he published a paper where they produced many more offspring. And these were all healthy offspring. So using the ideas from the mouse system, we went about developing an in vitro growth system for human oocyte development. And this required a multi-step system. So you couldn't just do it in one system. You had to change according to the stage of development, which David highlighted very well in his talk. So the first stage then is being able to get the primordial follicles to activate, to get them to start to grow. And then supporting the development of these isolated growing follicles. So maintaining the oocyte somatic cell interactions. And then of course, there's the final stage of oocyte development, and that's like looking at the oocyte and its surrounding granulosa cells. So as David said, the oocyte absolutely needs the connection between itself and the somatic cells. And then the fourth stage is to test the function of these in vitro grown oocytes in terms of their ability to resume meiosis after in vitro maturation and be able to be fertilized. And then, of course, to test whether any embryos that are produced are normal, looking at, epigenetics and metabolomics. So to take you through this, the system, this is a sort of piece, this is a small piece of human ovarian tissue, and it's really dense. And this has been donated for research, and this small piece could contain hundreds of oocytes. Now, we have to prepare the tissue in a way so that it will activate the growth of the primordial follicles that are contained within this tissue. And so if you just prepare this and just chop it up and put it in as dense cubes, then nothing will happen, or something might happen, but it will be very, very slow for it to happen. So what we have to do is prepare the tissue in such a way that we're pulling the tissue, that we're opening up, we're changing the architecture of the tissue. And initially, we didn't really know what we were doing with this, but we now know through the work of Erin Shui and Kawamura that the oocytes, what we're doing is we're interrupting the hippo signaling pathway, and we're able to activate these primordial follicles to start to grow. So this is what's happening here. You can see primordial follicles starting to grow here. And then they grow into multilaminar stages. You can see growing follicles, but you cannot leave them within that tissue. You have to take them out and then let them grow individually, because the follicles at that stage are communicating with one another, and they're inhibiting one another. So we isolate them, but not using enzymes. We isolate them by manually dissecting them using fine needles. We don't use enzymes because that damages these follicles. And then we culture them individually. We don't use a matrix to culture them in. They're cultured in V-shaped wells, and by doing that, they maintain their three-dimensional configuration. And these follicles are cultured in the presence of activin A, ascorbic acid, and a very low dose of FSH, but it's serum-free. It's a completely serum-free media. What we have observed is that we require the theca cells to be present for this culture. So we require some of the theca cells when we do the dissection, but there's an optimal amount of theca cells. If we have too many, we can really stifle the growth of the oocyte. So we have a kind of optimal amount of theca cells there. And this is what David was talking about. I mean, key to in vitro growth is maintaining the connection between the oocyte and its surrounding somatic cells. So making this unit, and that's what we found in collaboration with David, that the presence of activin and FSH also, a small dose of FSH, can maintain the stability of the cell-cell communications between somatic cells and between the oocyte. So we now know what some of the components that we need to put in the media to obtain good connections and the formation of antral cavities within the in vitro grown follicles. So if we can maintain those to the antral stage, then we have a big problem at the next stage, because we need to make a decision. And our decision is this: do we keep growing the follicle to produce a very, very large antral cavity, or do we focus on the part of the follicle that we are interested in? And we decided to do the latter. So when we have these antral follicles present, we've decided, okay, the whole large follicle really is, when it gets bigger and bigger, is really for an endocrine purpose. And what we're particularly interested in is the oocyte. And this is sort of similar to the sort of rationale that Epig used when he developed his system for the mouse model. So we want to focus on the oocyte and its surrounding granulosa cells, not the neurogranulosa cells. So we remove this complex and then place it onto a membrane, and we grow it for a further 2 to 4 days on this membrane. And then after that period of time, we then place the complex into maturation media, and we look to see whether these oocytes are capable of resuming meiosis. And remember, these have all come from the early stage of development, and they've grown within 19 to 21 days, and then they can go into maturation media. And you can see here that we do get maturation. We do get spindle formation, and we get polar body formation in these human oocytes that have been grown in vitro. We've also done this in the bovine, and we also get maturation of the oocytes after growth from the primordial stage. So we have this multi-step culture system that starts with the ovarian cortex, and we refer to as the microcortex once we prepare it. The follicles grow, and then we dissect them out, the growing follicles. We culture them individually until they form antral cavities, and then we remove the complex, and then we get them to mature. Now, at this stage, we haven't gone to the fertilization step because of licensing requirements there, but that's something clearly that we're working towards. But in healthy adult tissue that we obtain at the time of caesarean section, about 30% of the oocytes that complete the culture process can reach maturity. They can reach metaphase 2 stage. So, just to bring us back then, we've got this culture system. We can get these oocytes to metaphase 2, and we've been working on it for a very, very long time. So why pursue it? Well, the main reason that we are pursuing it at the moment is for basic scientific reasons, because we don't really know too much about the development of the human oocyte, and by having an in vitro model, we can learn so much more about it, and we can compare what we've found out from rodent systems to what we can find out from using a human system. But of course, there is a very, very practical application in terms of fertility preservation, and particularly for young girls, because young girls, their only option for fertility preservation is to store ovarian tissue. And if that ovarian tissue is not suitable to be reimplanted, then what happens? Then they have no options. It could, of course, be the next generation IVF if it would become efficient, and it would, you wouldn't have to use hormonal stimulation to obtain the oocytes. It would be very, very useful in animal production to increase the number of embryos from high genetic merit females, and also within wildlife conservation. So there are many, many aspects that are practical to in vitro growth. So it is important that it is pursued and that we can get to the next stage of moving towards applying it in a clinical sense. So what do we need to do then? So we've looked at various tissue sources, and we've looked at our healthy women, the tissue that we get from caesarean sections, and we can get metaphase 2 oocytes, as I've shown you. We've also looked at prepubertal girls, and I've also said that that's probably the most important patient group that would require this technique. But we're unable at the moment to get them to metaphase 2, and we're working on it. But we've done a lot of work, and we've published on this in human reproduction. And what we found is that in vitro, follicles grow at different rates depending on the age at which they came from. So the women between 25 and 38, then you can see that the rate of growth of these follicles in the green bar is, you know, quite high, but then it reduces in the younger girls between 12 and 15. And then when you go down to 3 to 10, it's very, very slow. So we now have to work out the tissue culture supplements that and the conditions in order to improve that growth. And is it timing or is it other factors? And that's a part of the work that we're involved in terms of moving this field forward. We've also looked at other tissues from Turner's patients and from chemotherapy treated groups. And of course, that's very variable depending on treatment and of course, depending on the type of Turners that we have. And we also have oocytes from gender reassignment group, and we're getting some promising results from the gender reassignment group, and we have been able to get metaphase 2 oocytes from that group as well. So there are some promising developments going on, but the main point is that with the culture system, one size does not fit all. The system has to be adapted for the tissue type and of course the patient group. So if we're moving towards an application, then we have to improve the culture conditions. And that's what David spoke about, having a basic understanding of all the factors that are responsible for oocyte and follicle development and cell-cell interactions, and knowing when to add these factors at the particular stage of development that's necessary. Now, when I showed you the metaphase 2 oocytes, I'm sure all of you noticed that these polar bodies were rather large. They're large polar bodies. And we don't know if that's an abnormality, but what we have been working on is changing that. And I can tell you that we've changed physical conditions and we've lowered our oxygen tension, and we seem to be having, you know, good preliminary results in terms of not seeing the large polar bodies. And in our bovine system, we don't see the large polar bodies. Another thing that I'd like to highlight is the timing of this growth. So David said that from estimates from Goujon, and we don't know exactly how long it takes a primordial follicle to go all the way to the point where it would ovulate, and when that oocyte has, you know, reached full developmental competence. But some estimate that it's about 100 days. Now, of course, I think most people would agree that it's not 100 days of complete growth, you know, continuous growth. It's probably a start-stop process. But we know we have colleagues who culture follicles under sort of slower conditions. Now, we wouldn't refer to our system as a fast-growing system. Other people would, but I would refer to it as a system where we take the brakes off the in vivo controls, and that's how long it takes. It can take between 21 and 24 days to get a fully competent oocyte or a meiotically competent oocyte. My colleague Helen Picton in Leeds has a system with sheep follicles where she grows them for much longer, for 36 days. Still less than in vivo, but what we now have a project in terms of comparing the slow growth system to our growth system, and to determine if the growth rate has an impact on developmental competence of these in vitro grown oocytes. So in the next steps then towards clinical application, we need to determine the health and developmental competence of these in vitro grown oocytes. We need to sequence them, we need to look at their epigenome, their metabolome, and of course, we need to fertilize them. We're working on an animal model, on a sheep and cow model, and we are hoping to have embryos and be able to get live young. We're a bit behind because of COVID-19, but hopefully next year we'll be able to say whether we can obtain sheep and maybe even cows from this system. And that would help us in terms of developing and pushing the human system forward. Now, of course, we have a long way to go, but there is a lot of potential for the in vitro growth system. And by our continuous efforts in terms of understanding the whole process of oocyte and follicle development, then I really think we will get there. So I'd like to acknowledge the people who do the work, and people and lab members, the nurses that consent the patients for us to get tissue, Professor Richard Anderson, who's our chair, and Hamish Waltz, the paediatric oncologist, and of course David Albertini, who's been involved in this work for a long time and has been essential for the development of the imaging techniques, and of course all the women who give us the tissue, the Medical Research Council and the Wellcome Trust who fund our work. And thank you all very much for your attention.
Well, thank you very much indeed, Evelyn. A very complimentary talk, and fantastic to hear two such world experts giving us their views on this really interesting topic. Perhaps just a quick question for yourself, Evelyn, that's come up in one of the questions that the audience have posted, is you highlighted the role of activin in your culture system. Have you explored the role of the IGFs as well in this system?
Yeah, not in the human model, but in our cow model, we've looked at IGFs and the IGF binding proteins. And of course, the cow is a bit different from humans. But yeah, IGFs can be in the second stage of the culture system. The IGF, the presence of IGF-1 can be beneficial in terms of getting to the antral stage, and also in terms of maintaining the interaction between the oocyte and the somatic cells. But I think it changes, as David said, it's a moving target. So it's really getting to the point when we know when to add it and when to take it out.
Thank you. And can I ask David a question on your use of these animal models? So clearly there's no substituting human tissue where possible, and you highlight the large animal species as well. David, do you think the rodent's follicle is really a useful substitute here, or are there sufficient species differences that you really need to use a monoovular species to really model human ovaries?
Fair question. I think there are enough differences in the rodent models to preclude them from us moving the clinical field forward. I think now that, as we've discussed, people like yourselves and so many others, have banked so much tissue, and we're, I believe, on the road with, not only the work that's being done in the human, but we have our colleagues that have done some elegant work in the primate as well. I don't honestly see a clear utility for the rodent models at this point for optimizing IVG, especially for oocyte production, quality oocyte production.
Thank you, David.
You're welcome.
We've had a question for Evelyn, asking whether you've had the opportunity to look at the chromosome status of any of the mature oocytes that you've produced and their complementary polar bodies.
No, so we're currently, we have a project. We're in collaboration with cell biologists that work on meiosis, and so we're in the process. We will be looking in great detail at these at the chromosomes and of course the polar bodies as well. But that's work in progress.
Thank you, Colin. Did you want to chip in with a question?
Yeah, I wanted to come back and ask a question, but it can be either David or Evelyn. But you know, the fascination around what David was saying, that there's this continuous cross-talk between the cumulus cells and the oocyte, how important that is. Is there evidence? I mean, does that communication, is it there all the time, even from the moment that you have a primordial follicle and you have all those flattened pregranulosa cells all stuck around the oocyte? Is it always talking?
Yes. So it's been some time actually since you know, ultrastructural studies were done. Actually, and the initial studies were done in the fetal rabbit ovary, where you can follow the transition from the cyst with all the oligonia in it to primordial follicle isolation, the breakdown of the cyst or the nest, and functional gap junctions were demonstrated, both in terms of dye transfer and in terms of sort of meeting all of the then morphological requirements that would demonstrate that. And so now you can imagine, since then, you know, the work has been extended to actually knocking out the genes for the membrane proteins that mediate those forms of communication with consequential disruption. And it's interesting, there have actually been a few papers now. There's this great interest in how our environmental factors or exposures during pregnancy possibly influencing the development of the ovary of the testis and the ensuing germ cells. And some of the environmental toxicants that have been looked at, which happen to be estrogen mimics like bisphenol A, seem to target this communication pathway and be disruptive, you know, be disruptive to it. Now, the question as to, you know, how long do these persist for? And as far as we know, they only become more and more prominent. And now we're just talking about one class of interaction, okay, which are, say, gap junctions. But as I mentioned, there are about 7 or 8 different types of signaling mechanisms now, and they come on or come off at different times. The direct ones that really allow for metabolic cooperation are persistent throughout the course of folliculogenesis.
Right. Wow. Okay, that's great. Thank you very much indeed.
Thanks. Can I ask a question for both of you again, perhaps? And I suspect perhaps both of you will have an opinion on this. David, you highlighted the fantastic work coming from Japan over induced pluripotent stem cells to make oocytes ultimately. One of our audience questions whether we need to go back that far, or whether there might be stem cells within the ovary that can serve a function along those lines.
Well,
yeah, I think Evelyn would be better suited to address that.
Yeah, well, I mean, our work shows that there seems to be a population of cells within the ovary that has the potential, under certain conditions, to form what appear to be oocytes. So, of course, we still don't know enough about them. But yeah, I mean, there's a lot of evidence now to suggest that these cells, under the right conditions, could form what appear to be oocytes. So again, it's the same problem. We have to work on the conditions to, you know, activate them to become oocytes, and then to test their functions. So maybe we don't have to start with induced pluripotent stem cells. But if we do start with induced pluripotent stem cells, we have the same problem as the in vitro growth from the primordial follicle, because you have to have the culture systems to support the growth of a newly formed primordial follicle. So regardless of where you start in the process, you still need these in vitro growth systems. And it would be combining, say, the work that's been done on the people that are working on the stem cell field up to the point of, say, producing primordial germ cells, and then getting them towards oocyte development, mixing that with in vitro growth systems to get developmentally competent oocytes.
And I think just to add to Evelyn's comment, you know, among the breakthroughs that have come from at least one of the Japanese groups, and I believe it was the Saito group originally, was to recognize the importance of matching the germ cell with the correct somatic stage of, in their case, they realized that it had to be, I believe, newborn day 2 stromal cells from a mouse ovary that would best match with their PGC-like cells and allow for reconstitution of effective follicles. And so this again tells you that this moving target, that is indeed oogenesis, is changing the way that it communicates with its somatic environment. Okay, and that the IVG models that Evelyn and others are developing are somehow going to have to factor that into the equation as the recipes become perhaps unique for the source of the germ cell or the source of the material from the patient moving forward.
Absolutely, and I think also in terms of the Saito's studies, you know, when you have to match the somatic cells, then you have problems in terms of an application. But with the cells, if there are cells within the ovary that will develop towards oocyte development, there also has to be somatic stem cells. And so I think the study of the ovary in terms of finding this, you know, these populations and working out how you can activate them will be really important.
I've got a question here. Well, it's a comment really from, this is for Evelyn, from Laura Ball from Helen Picton's lab in Leeds. And she's saying that, you know, I really do understand the desire to reduce the time follicles spend in culture. Is there a possibility further that follicular genesis could be completed in less than what you are currently reporting? What's your thoughts on that, Evelyn?
Yeah. Sure. Well, we never set out for a particular time. This happened. So when we took it out of its environment and the way we manipulated the follicle, that was the time that it took. So, but I do think there would be different times depending on the stage of development that we take the follicles from. So we can see that in the prepubertal girls. So I think in Helen's lab, of course, they're trying to recapitulate a whole large follicle and keep it going. And we're not doing that at all. We're focusing on the oocyte. And I think that when we take all the breaks off, that the oocyte does develop much more quickly. And when we've looked at this in cow, in pig, in sheep, in a whole range of species, they actually form a very, very similar trajectory. The mouse is the fastest, but it's the fastest in vivo. But all the other species seem to do it within this time frame, which would suggest that there is, you know, a kind of default pathway. Now, whether that's fully functional or not, that's what we hope to find out.
Okay, thank you. Richard?
Yeah, so Evelyn, you highlighted the importance of the source of your material, of differences between prepubertal girls, adults, other sources. We've got a question on, you mentioned the transgender patients, and there's a question about whether there's relevance to the gender affirming endocrine treatment they would have had. Has that had an impact on these ovaries?
Yeah, so that's absolutely what we're studying. I mean, Richard and I, we have an MD student and we've had a PhD student who have been working, looking at the gender reassignment ovaries and looking at how the testosterone affects the primordial population, its ability in terms of DNA damage, DNA repair, and it's, you know, how effective it is in terms of, or how effective the primordial follicles are in terms of activating and growing. So at this stage, we don't have, you know, it's early stages of the project, but we're certainly looking at that.
David, do you think that testosterone is a very interesting molecule, and you know, obviously having a lot of clinical application these days in IVF for activation of follicles, enhancing growth? What do you think about the use of chemical agents to activate follicles for in vitro growth? Is that an appropriate way forward?
I think that the database that our current understanding is built on is pretty solid in terms of you know, I think we're exploring mechanisms that are in fact not that unique to the ovarian follicle activation. I think you know, as many have referred to, this hippo-YAP pathway is a constituent pathway for lots of cells. And in fact, you know, I think some of this is not only uncovering for us what the, if you will, silencing system is that's inherent to the structure of the ovary for maintaining the primordial follicles in that state. But I think it's telling us something about the aging process too, and something that you know, in theory, you as a clinician might be able to use for patients that are showing POI, or you know, you've got, I presume you've got some mosaic Turners on your hands, for example, and you know, I definitely think that we have a pharmacopoeia right now that is working in animal models, and you know, is working. It's worked in your hands. I mean, you guys have published some of this work as well, using the inhibitors and activators of the follicle. So I say go for it at this point. You know, it'd be different, I think, if we did not see that there was widespread conservation across different animals, where being able to do the targeted genetic manipulation, as we've been able to do in the mouse, people like Quilu and others, okay? You know, I think the evidence is pretty good that there's some translational value in the human, and so I think it's an important building point for us.
I would introduce a note of caution with these inhibitors, because when we've used them, yeah, of course you do get an increased activation, but the quality of the oocytes when you grow them for a further period of time is poor. So, and also we've looked at DNA damage and DNA repair mechanisms, and they are in fact compromised when you use these pharmacological inhibitors. So I think we really do need to have a greater understanding of using the pharmacological inhibitors to increase activation of primordial follicles.
Well, I must say, I believe Richard set me up on that one. Because
think
had
it
I
does.
looked back, because I know your paper, and that was exactly the take-home message. And, you
But
know,
never
had I
give up.
recalled it a little
Right.
better, I might not have been tricked into the answer that I provided
Okay.
for our audience. But that's what friends are for.
Okay,
Yeah,
great.
a little bit of comment there, perhaps.
Okay, I
Take
think
Ken's
we're now
you really see this.
hello, Richard.
Yeah, I think we lost him.
We lost you, Richard.
Yeah.
Okay.
I lost
Being punished.
I was just going to ask, yeah, but I was going to ask the speakers just to give a very short answer to how they might see this being implemented in practice and which they would want to see this first applied for, assuming, you know, it works and it's safe.
Well, I mean, the biggest application would be for the prepubertal patients that cannot have transplantation.
I would, you know, having spent 5 years in the Center for Human Reproduction, where the average age of our patients is 42, and seeing so many of the POI patients coming in, I see that population of women as if you could store the tissue soon enough, they would have an immediate need for available oocytes using this kind of technique.
Thank you. So you're both coming at it then from the opposite ends of the age spectrum, the prepubertal to the older. So a broad range. So fantastic. Thank you both very much. Perhaps I could hand back to Colin for a few final words.
Thank you very much indeed, panelists, and thank you again to Richard Anderson for moderating that session. It really was a really fascinating talks that we've had today. Again, I just want to thank everybody very much for all the questions. We still have some which we will try later to finish off with the panelists and try and get some sort of a consensus on some of the ones we couldn't cover. But it was a wonderful session. Thank you very much. And I just want to remind everybody that there will be another session this week on Friday. This is on quality control part 1. And we really do look forward to hosting more of these really high quality sessions for all of you out there in the IVF and reproductive fields. So again, thank you very much, David, thank you very much, Evelyn, and thank you very much, Richard, for all of the work and the moderation and putting this whole session together. Thank you.